Hello, my name is Yoo-Jung Kim, and this is my presentation for the article Increased Tumor Homing and Tissue Penetration of Filamentous Potato Virus X. Viral nanoparticles are an exciting new potential drug delivery method that is both safe and versatile for humans. They can be engineered with other ligands, proteins, and molecules to deliver a drug and target it to a specific tissue, making it ideal for treating diseases such as tumors and cancer. Additionally, the specific viral nanoparticle used in these applications are derived from plants and are non-pathogenic to humans, making it safe for human use. Targeting tumor cells and loading chemotherapeutic drugs onto or inside these viral nanoparticles have allowed higher accumulation of drug inside tumors, thus decreasing side effects and increasing therapeutic efficiency. Recent studies suggested longer particles with higher aspect ratios are able to home in onto tumors more effectively and have better pharmacokinetics. However, the majority of nanoparticles being investigated for drug delivery or platform technologies are low aspect ratio particles such as spherical micelles and dendrimers. Carbon nanotubes are also being investigated and have high aspect ratios, but the drawback is that they have very low biocompatibility. One of the reasons why there are so few high aspect ratio nanoparticles being investigated might be because it is extremely difficult to create them synthetically because of polydispersity and poorly controlled chemistry. In this paper, Shukla et al. Did a side-by-side comparison between a spherical low aspect ratio viral nanoparticle called cowpea mosaic virus, or CPMV, and a filamentous high aspect ratio viral nanoparticle called potato virus X, or PVX. Using PVX is beneficial because the structure is known, it is safe for humans, and as discussed earlier, can be conjugated with different ligands for diverse applications. Both of the nanoparticles were pegylated to prevent immunogenicity, decrease nonspecific interactions, and increase circulation time in plasma. This first image shows the characterization of the two particles, indicating the molecular weight and zeta potentials. In order to view the particles, Shukla et al. Attached a fluorescent moiety onto the viral nanoparticles. This is also done in subsequent assays in this study. The second image shows the uptake of the viral nanoparticles into tumors. Both viral nanoparticles were able to target and home into the tumors comparably. However, significantly more PVX accumulated in the core of the tumor compared to the CPMV. Additionally, PVX seemed to be more distributed throughout the tumor compared to CPMV, which tended to clump together. The third image shows the biodistribution and targeting abilities of both particles in vivo. In A, we see that there is enhanced retention of PVX in more tissue compared to CPMV. In B, we see that there is significantly more PVX in specifically tumor tissue compared to CPMV, suggesting improved targeting ability of PVX. Additionally, in C, we also see that there is more PVX in tumor tissue compared to the CPMV. The figure on the left shows the plasma circulation time of both PVX and CPMV particles in plasma. PVX has a half-life of 12.5 minutes and seemed to have lower plasma circulation time when compared to CPMV with a half-life of 20.8 minutes. This is surprising because initial hypothesis proposed that longer filamentous particles will have longer circulation time, which is what leads to better pharmacokinetics. However, these results may indicate that perhaps the increased pharmacokinetics of longer particles is not because of longer circulation time, but because of something else. The figure on the right shows the biodistribution of fluorescently labeled CPMV and PVX in different organs of mice. The results of this experiment shows that there is increased accumulation of PVX in the spleen and increased accumulation of CPMV in the liver. When the data is normalized to the samples that received only PBS, we still see this trend. In conclusion, Shukla et al. Were able to provide evidence that PVX, a higher aspect ratio nanoparticle, had increased targeting and homing abilities, and improved biodistribution compared to CPMV. As a result, they showed that PVX may be a promising new method of drug delivery for cancer therapeutics. Continuing in this project, Shukla's group may find it interesting to determine what exactly is the mechanism that increased biodistribution for PVX, since it was not the circulation time. Additionally, although the drug may be conjugated onto the nanoparticle, it is also important that the drug is released in order to elicit therapeutic efficiency. Additionally, they must also test the stability of the drugs after they are conjugated onto the particles. Therefore, the next study they should do is to load up the viral nanoparticle with chemotherapeutic drugs and see their release profiles, kinetics, stability, and efficiency in vivo tumor models. Lastly, it would be interesting to see other pathologies this technology can be applied to, and as such, perhaps the group can do a survey of other diseases that may benefit. Thank you.